So good afternoon, everyone. My name is Paulina. I'm a second-year PhD student at the University of Warsaw. And today I will be presenting this talk on behalf of my, on behalf of my supervisor, who unfortunately couldn't be here today. So I'll be talking about GPCRVS, which is a web service that our lab has recently developed. And GPCRVS uses two machine learning algorithms in combination with structure-based virtual screening to assess compound activity against a number of G protein coupled receptors. However, before I get into the specific features of GPCRVS, I would like to give you a bit of background information on machine learning, artificial intelligence, drug discovery, and G protein coupled receptors. So virtual screening is a drug discovery technique that is used to identify potential active compounds from large compound libraries. It can evaluate the affinity of millions of compounds and produce only a few hundred that have the best affinity towards a target. And these few hundred are then tested in assays, and this saves both time and money. There are two main kinds of virtual screening. There's structure-based virtual screening and ligand-based virtual screening. So for structure-based virtual screening, we need to have the structure of our target, as this involves molecular docking, which assesses how well a compound fits into the active site of our target. Ligand-based virtual screening, on the other hand, it requires information on other ligands that are known to bind to our target. And based on this information, it can then find new compounds with similar chemical properties. In GPCRVS, ligand-based virtual screening is implemented through a neural network in TensorFlow and a gradient boosting machine in LightGBM. So artificial neural networks are computational models that are inspired by the structure and function of the human brain. Sorry, I think I'm having some issues with the presentation. No, but the presenter mode isn't, it keeps skipping. The text keeps going over to the text that's for a different slide. This is for the last slide, and it keeps showing 18 out of 18. I'm not entirely sure. And it's fine for 2 seconds, and then it just ends up skipping for some reason.
I don't know. I don't think there's a solution.
Okay. Right. So neural networks are mostly used for pattern recognition, clustering, and classification tasks. And they excel at tasks like image recognition or natural language processing, making them a cornerstone of modern artificial intelligence. Neural networks are composed of nodes, interconnected nodes, which model neurons. And these nodes are connected to each other by edges, which model synapses. They're composed of an input layer, one or more hidden layers, and an output layer. So signals pass from the input layer through the hidden layers and towards the output layer. And what happens is that each neuron, it receives an input, and it processes that input using a weight sum, applies an activation function, and produces an output, which is then passed on to the neurons in the next layer. The training for these networks can be either supervised, where the model is trained on labeled data, or it can be unsupervised, where the data is unlabeled and the model has to find patterns and relationships on its own. Neural networks consisting of multiple layers with complex architecture are called deep neural networks. There are numerous types of neural networks. There's feed-forward neural networks, which are used for standard and classification and regression tasks. There's convolutional neural networks, which are used for image recognition and computer vision applications. Recurrent neural networks, which are suited for sequential data like text. Generative adversarial networks, which can be used to generate synthetic data. And transformer networks, which can be used to capture global dependencies in sequential data. So TensorFlow and PyTorch are two very popular open source machine learning frameworks that can be used to construct neural networks. TensorFlow was developed by Google Brain, and it's known for its scalability and the fact that it supports many different platforms, including desktops, servers, and mobile devices. It also boasts strong built-in visualization capabilities, as well as a comprehensive API ecosystem, which facilitates seamless integration into production environments. In addition, it has a static computational graph, which provides the advantage of faster and more efficient training through parallelism. PyTorch, on the other hand, was co-developed by Adam Pashke, who is coincidentally a graduate of the University of Warsaw. It's known for its user-friendliness, partially due to its integration of the Python programming language. And in contrast to TensorFlow, it has a dynamic computational graph, which allows for intuitive model building, as well as interactive debugging. And this is very useful for rapid prototyping. The GPCRVS neural network is implemented in TensorFlow. So the second machine learning algorithm that's implemented in GPCRVS is based on gradient boosting decision trees. And this is a machine learning technique that is used for regression and classification tasks. A model is built by sequentially combining multiple weak learners, usually decision trees. And the idea is to iteratively improve the model by focusing on the errors of the previous model. And this is achieved by training each new model to predict the residuals of the combined ensemble of the previous models. So the process begins with an initial prediction, and then a series of decision trees is built. And each tree is trained to predict the residuals from the previous step. These predictions are then added together, each with a weight that is optimized to minimize the loss function. And then this continues until the model reaches a specified number of iterations or until the error converges. Gradient boosting is, in general, very accurate and able to handle different types of data, but it's prone to overfitting. It requires the careful tuning of parameters such as learning rates, the number of trees or tree depth, and overfitting can also be mitigated through the use of regularization techniques. Gradient boosting can be implemented through frameworks such as XGBoost or LightGBM. LightGBM is short for Light Gradient Boosting Machine. It is a gradient boosting framework that was developed by Microsoft. It's optimized for speed and performance, and it's known for a reduced memory consumption. It also boasts two novel techniques that allow for a faster processing, and these are gradient-based one-side sampling and exclusive feature bundling. Gradient-based one-side sampling enhances efficiency by prioritizing data instances with larger gradients, as these are more informative. And in this way, it reduces the number of instances required for training while not affecting accuracy. Exclusive feature bundling improves computational efficiency by reducing the number of features. So this is done through the bundling of mutually exclusive features, which reduces the dimensionality of the data and the complexity of our model. Both of these methods decrease memory usage and enhance efficiency. Moreover, due to LightGBM's support for parallel computing across multiple processors, distributed systems, and GPU architectures, it can efficiently handle very large data sets while ensuring efficient scaling and maintaining a high performance. These are the features that we use LightGBM these features are the reason why we use LightGBM to implement our second machine learning algorithm. Two other popular frameworks include the aforementioned XGBoost or extreme gradient boosting, as well as CapBoost. XGBoost is known for its high computational efficiency, its scalability, and its ability to handle complex relationships and data. CapBoost, on the other hand, is unique in its automatic handling of categorical data, and which reduces the need for extensive preprocessing. Now, moving on to the more biological aspect of our research, G-protein coupled receptors, or GPCRs, constitute the largest group of cell surface receptors, and they regulate the majority of physiological processes. They're composed of an extracellular N-terminal, 7 transmembrane alpha helices, an intracellular 8th helix, and an intracellular C-terminal. They transduce signals through either G proteins or beta arrestins, and ligands can bind to either an orthosteric binding site, which is the main binding site located towards the extracellular site of the receptor in between the alpha helices, or an allosteric binding site, which is where substances called modulators bind, and they induce conformational changes in the receptor, which can either inhibit or enhance its activity. GPCRs are of particular interest in pharmacology since they are the targets of more than 38% of the drugs that are currently available on the market. There are also certain GPCRs that are classed as immune receptors, which are responsible for regulating adaptive immunity, and thus they represent important drug targets in new antiviral, antimicrobial, and immunosuppressive pharmacotherapies. So we first tested our machine learning algorithms on chemokine receptors, and chemokines are small proteins that are known to participate in the transport of leukocytes to injured areas of the body. They're known to play a role in numerous pathological processes, and because of this, their receptors are promising drug targets for the treatment of conditions such as inflammation, allergies, autoimmune diseases, vascular diseases, and allograft rejection, for example. So we performed ligand-based virtual screening for chemokine receptors CC1R through CCR6, as well as CXCR1, CXCR2, and CXCR3. And we combined these two machine learning methods with structure-based virtual screening and molecular dynamics. And thanks to this, we were able to propose novel active compounds for CCR2, CCR3, and CXCR3. So our machine learning models were trained on data sets obtained from Campbell. These were IC50 data sets. The activity prediction was based solely on Smiles-based fingerprints called extended connectivity fingerprints. The light GBM predictions were in general found to be more accurate when the inactive compounds were not included in the training sets, which you can see in the plot on the upper right. The predicted activity values were highly accurate, which you can see by comparing the blue dots to the red trend line. And in the bar graph below, you can also see that the number of correctly predicted compounds was also higher when in comparison to the model trained with the inactive compounds. So what this means is that for multi-class classification tasks, the removal of negative data points improved the prediction, while the opposite was found to be true for binary classification, which just distinguishes between inactive and active compounds. Light GBM provides continuous activity values in contrast to the discrete ranges of activity values provided by the neural network. In tests performed for the chemokine receptor CXCR1, you can see that the activities of known drug candidates were predicted more accurately by the neural network, while the opposite is true for CXCR2. Compounds that had a p-Campbell value of above around 5 were also predicted more accurately. This is the main page of our web service. It contains the form for uploading the compound that users want to test. So what you have to do is you put in your name, your email address, and a job description that will allow you to recognize the job you submitted. You can choose whether you want to obtain results for the orthosteric or the allosteric binding site, and you can upload a ligand that is either a small molecule or a short peptide up to 6 amino acids. And these can be uploaded in a number of formats, including SDF, PBD, PDB, QT, Smiles files, and FASTA files. Most of the receptors included in GPCRVS are Class B receptors for peptide hormones, but the aforementioned chemokine receptors are also recently added. Most receptors are present in both an active and an inactive state, depending on their availability in the protein data bank. The compound binding mode for each receptor is provided and shown with lick plots. The predicted activity values, the predicted activity of the tested compound is shown as a red line, and this is juxtaposed on top of the experimental activities of known compounds for a certain receptor. So here, the results obtained for Maraviroc are compared to those obtained for 4-ethylaniline. So the main aim of GPCRVS is to discard inactive compounds. As you can see here, both machine learning algorithms correctly predicted that Maraviroc is active for the CCR5 receptor, while 4-ethylaniline is not. Similarly, the binding affinity estimated by Autodoc Vina is also much lower for Maraviroc. And this is, yeah. GPCRVS also estimates the overall level of activity for a selected compound. So compounds can be classified as either as compounds with a high, low, or medium activity. Maraviroc was classified as a highly active compound by the light GBM model. The red line corresponds to the predicted activity of Maraviroc. The blue points correspond to the predictions for known ligands of CCR5. And on the x-axis, you can see a ranking of these compounds obtained from assays. And you can also see here that Maraviroc is classified as one of the most active compounds, ranked as first or second in the functional assays. So in contrast, it should be predicted as inactive for other receptor types, which when tested for CCR4, both the machine learning models correctly predicted that Maraviroc will have a low activity value. Autodocvina was found to not distinguish as well between receptor subtypes, as the estimated docking score for Maraviroc is only slightly higher than that for CCR4 than that obtained for CCR5. An overview of the compound activity predicted for all the GPCRs is presented in a compound activity profile. And on this graph, we can see the TensorFlow predictions in gray. These also correspond to the height of the columns. The light GBM predictions are presented in black, and the Autodocvina score is presented as the color of the columns. So under the overview plot is a collection of collapsed tabs. After expanding them, you can view detailed results. So before I end my presentation, I'd like to acknowledge the people who contributed to the development of GPCRVS the most. And that would be our group leader, Dorota Lepek, who was responsible for the architecture, backend, and front end of GPCRVS, as well as algorithm implementation and parameter tuning. Matt was responsible for testing the machine learning modules in terms of dataset composition. He also made a preliminary sequential model of a neural network, and together with Szymon and Sławomir, he also curated training datasets obtained from Campbell. Kushil and Przemysław made the web framework, the visualization of results, and integrated the backend with Apache. Kavita made the graphics for the website, and I was responsible for the module that made it possible to include peptide compounds in GPCRVS. I also made the compound activity profile and performed tests. I'd also like to thank the National Science Center in Poland for funding this research, and PL Grid for providing computational resources for the MD simulations, and the Faculty of Chemistry for providing the infrastructures for GPCRVS. Thank you. I'll be happy to answer questions.